Now we will be discussing about the platelet count. So what will be our learning objective? At the end of demonstration, you should be able to interpret the result of platelet count and set the normal value of platelet. So method again, we will use Newbar chamber, of course improved one. Indirect method can be from peripheral blood smear. Platelet count is made relative to RBC. It is indirect method. And third will be automated. Automated hematology analyzers are also determining platelet volume, which has been correlated to several diseases. In general, mean platelet volume has an inverse relation to platelet number. Now, reticulated platelets are new released platelet that retain what? Residual RNA, which is analogous to what? Red cell reticulocyte. Reticulated platelet count give an estimate of thrombopoiesis and may be useful to distinguish platelet destruction syndrome from hypoplastic platelet production. Now, indirect method. So, make a peripheral blood smear. How? Prick finger after placing a drop of 3.8% sodium citrate on fingertip. Why? To avoid the platelet aggregation. You have to make peripheral blood smear, but when you are pricking the finger after placing a drop of 3.8 sodium citrate on a fingertip, so that there should not be any platelet aggregation. And again, stain the same Lisman stain. Point number 1. Second step, reduce the field of eyepiece as we do in reticulocyte count. So you have to reduce the field of eyepiece. Count till 100 platelets are found in different field. Calculation is very simple. Number of platelet in N field will be 100. Total number of red cell in N field is equal to Y. So platelet count, you can count like that, 100 upon Y into RBC count. But you should know number of RBC. So this is the only shortcoming, that you should know RBC count. And in severe anemia, sometimes there is falsely elevated platelet if you are using indirect method. So that can be a drawback. Otherwise, we can use indirect also. Now, direct platelet count. What is the principle? Principle is same as we have done with the RBC and WBC, that we will dilute to the known amount and number of platelet are counted in a known volume. After that, we will multiply by a multiplication factor. Apparatus are same, improved Newbar chamber, compound microscope, RBC pipette we are using. Okay, platelet diluting fluid, disposable blood lancet, spit swab and cotton cloth. Now here again they will ask you, like there was a Turk fluid, there was a Ham fluid. Here for platelet, which diluting fluid we use in our lab? Detergent 1% for hemolysis of RBC, sodium citrate 3% as an anticoagulant, formalin 1% as well as NaCl 1%. What other fluid that can be used? Sometimes they ask you, very famous Rees-Aker fluid. It has brilliant crescile blue which stain the platelet, 3.8% sodium citrate as an anticoagulant, 14% formalin as RBC preservative and distilled water. Procedure just like RBC and WBC. Take adequate diluting fluid in a watch glass, prick ring finger under aseptic condition, wipe off the first blood drop, suck again the diluting fluid up to which mark? Again 0.5 mark, to coat the wall of RBC pipette with anticoagulant. Now suck the blood to 0.5 mark, the total 0.5 diluting plus 0.5 will be the blood. Now clean the tip of pipette, suck diluting fluid till solution of 101 mark, because this is just like RBC. In RBC, we are using 101.5 and then 101. Similarly, we are using 101 mark. Now mix by rolling the pipette in between the palms. Keep the pipette for 10 to 15 minutes. Place cover slip over the chamber. And now you have to focus on what? WBC square or RBC square. Just like in RBC, we have to focus on RBC square under low power and remove the chamber without disturbing the focus. Similarly, charge the chamber after discarding 2 drops from that stem. Wait for 2 to 5 minutes for platelet to settle down. Then fine adjustment is done in low power, but you have to focus on high power. Then count platelet in RBC square. Similarly, all 25 medium square is preferred to reduce error. Now again we have to follow the inverted L I have discussed. Now calculation. You have to calculate. Calculation here, dilution factor is how much? Dilution factor is 200, but final multiplication factor, if you counted in all 25 RBC square, it will be 2000. So this is almost similar, just like we have done in the RBC, but the only few things are to be noted here. Just remember those points, they can ask you in viva. Thank you.